In the previous lesson on the origin of life, we looked at the top-down strategy, where we study bacteria that live in extreme environments, for they might share characteristics with the earliest bacteria. In this lesson, we'll look at the bottom-up strategy. Here, we look for places on Earth where energy and raw materials would have been available to produce biomolecules, and then later organize them into cells. Now, this picture is an artist's rendition sort of the flow of time from the early Earth to a planet teeming with life over here on the right. Notice in the ocean here we have simple molecules, perhaps made in the atmosphere, gases interacting chemically in the atmosphere, maybe powered by ultraviolet light as a source of energy, or lightning, etc. But here we have simple molecules, and as we move to the right we get more complicated molecules, and then we start to have the first living cells. And these cells then would undergo evolution by natural selection, evolving into more complicated cells, and then multi-celled animals and plants and such, and then colonizing land, and we get the millions and millions and millions of species on Earth today. So this is the story that modern science presents to us, that the Earth began as a lifeless rock, and now there are countless species. The answer to that puzzle, how might that have happened, is we have to understand how the first living cells emerged on Earth. And from there, we have a good understanding of how evolution might work. So the strategy for the bottom-up strategy is to try to find places on Earth where the right kind of chemistry was happening to produce the right sorts of organic molecules, and then how they might have assembled into the first cells. Now, one example of a bottom-up strategy is what we're going to call the primordial soup hypothesis. This was an attractive idea in the 1950s, and the idea is this, that in the atmosphere there would have been various gases in the Earth's atmosphere, and they might have undergone chemical interactions, perhaps powered by ultraviolet light or lightning, and new organic molecules then would have rained down into the oceans, and the oceans over time would have become more rich with organic molecules, and perhaps somewhere in the oceans those organic molecules assembled into the first cells. We'll call this the primordial soup hypothesis, that the oceans were enriched with organic materials, and chemical evolution ultimately led to the emergence of the first cell. Well, one of the early advocates of the primordial soup hypothesis was Stanley Miller. And in the 1950s, he conducted an experiment to test the primordial soup hypothesis. He wanted to simulate the conditions on the early Earth, and he did so with this apparatus over here. He had a chamber that he filled with water to simulate the oceans, and he had a chamber here to simulate the atmosphere. So he put various gases in this chamber here. So he had ammonia and methane and hydrogen. Notice he did not put oxygen. Scientists then and now do not believe that the early Earth had oxygen in the atmosphere. As a source of energy, he put these electrodes here that would generate a spark to simulate lightning or ultraviolet radiation from the sun. Now, whatever molecules would be made here would be condensed down back into the ocean, and then evaporation would happen here, and we'd get more chemistry, and so the apparatus would cycle like this. Miller wanted to find out whether any interesting chemistry might have happened. Could you take simple molecules in the atmosphere and turn them into the molecules that we find in cells? Surprisingly, after a few days, the solution turned brown. And when he analyzed the solution, he found amino acids and all kinds of other organic molecules. Amino acids are the building blocks of proteins. Now, he didn't make a living cell, but he did make a whole bunch of organic molecules that are found in living cells. This was very exciting to scientists at the time, and it gave support to the primordial soup hypothesis. However, there were some criticisms that emerged over time. For one, in the 1960s, NASA scientists studying nearby planets like Venus and Mars were able to determine that the atmospheres of those planets did not have the kind of mixture that Miller put in his chamber. For example, there's a lot more carbon dioxide in the atmospheres of those planets. And if Earth was anything like Venus and Mars, then perhaps Earth had a lot of carbon dioxide in its atmosphere as well. And so the criticism for Miller's experiment then is that he used the wrong mixture of gases here. For when you start to take out some of the hydrogen and the methane, for example, and replace with carbon dioxide, you get much less of the production of those interesting organic materials. So that was one criticism that arose in the 1960s. In addition, some scientists began to doubt whether the primordial soup hypothesis would even be a workable solution. After all, the ocean is vast, and any organic molecules that would be dissolved in the ocean would sort of just float away, and it would be really difficult to get high enough concentrations of these things to make them interact to form even more complicated molecules, resulting in the first cell. So the primordial soup hypothesis began to have its critics. So the search continued. Are there places on Earth where chemistry might occur and concentrate the resulting organic molecules? In the 1970s, scientists using unmanned submersible submarine type things were studying the ocean floor. In particular, they were interested in volcanic activity at the bottom of the ocean. They discovered various kinds of volcanic features called hydrothermal vents. Now, there are different kinds of hydrothermal vents, and we'll be looking at alkaline hydrothermal vents. This is a picture of some of the rocky structures near an alkaline hydrothermal vent. Now, these vents are regions of volcanic activity on the ocean floor, and so the vent has a constant supply of energy and simple molecules from the Earth's interior. So there's hot water coming up through these rocky structures, and that hot water contains various gases. And the rock, of course, is composed of various minerals, and then the ocean water has various gases dissolved in it as well. But interestingly, if we look at the microstructure of these vents, there are very small chambers throughout the vent. And so these small little chambers would be capable of concentrating any organic molecules that might be produced in these chambers. They would be trapped in these chambers, able to interact with other molecules being produced in the chambers. And so this was an exciting discovery. Perhaps here we have an environment where new organic molecules might be made and concentrated sufficiently so that further chemical evolution might happen. In this diagram, we'll look at a cartoon version of an alkaline hydrothermal vent. Here we see the vent structure here, and we have hot water with various gases and such percolating up through these small rocky compartments. Notice this is not Darwin's warm little pond. After all, Darwin's warm little pond would suffer the same criticism as Miller's primordial soup hypothesis. Here we have hot fluids percolating through tiny, tiny little compartments. Now we'll take a magnification of one of these chambers. Here we see it up here. And with carbon dioxide and the ocean water and a source of hydrogen from the vent and a source of nitrogen from the environment, we have the elements that we find in living things today: CHON, carbon, hydrogen, oxygen, and nitrogen. And the idea is these simple molecules will undergo chemical reactions to form more complicated organic compounds. This would be chemical evolution. In addition, the minerals that make up the walls of these chambers have the capacity to speed up chemical reactions, so they catalyze chemical reactions. And so here then we have a promising location for the origin of important biomolecules found in living cells. In fact, we might say that the alkaline hydrothermal vents may have been a nursery for the formation of these biomolecules and later free-living cells. So it's an interesting analogy. It's a powerful analogy that life on Earth may have taken hold right here in these tiny little rocky compartments. Now, there's a scientific consensus in the steps to the formation of life on Earth. First, you have to make these biomolecules, and then they have to organize into cells. So we'll take a look now in this region here. We'll look at some of the biomolecules that are sort of currently found in cells, and then we'll see how they are sort of organized into a cell. Here are four classes of molecules that we find in cells. For example, we have fat molecules. We have sugar molecules, like glucose. We have amino acids, the building blocks of proteins. And then we have these nucleic acids. These are going to be the building blocks of DNA and RNA. So the idea is that in these tiny compartments, the chemistry was going on here in such a way as to produce the very kinds of molecules that we find in cells today. But how do these molecules organize into a cell? Well, let's take fat molecules, for example. They have a very interesting property. When you take a lot of fat molecules and put them in water, they tend to associate. The tails of the molecule tend to associate with each other. They don't like water, in a sense. And if you have enough, a highly concentrated bunch of fat molecules, then what they do is they assemble into these membrane-like structures. Well, these are precisely the membranes we find in cells. Cell membranes are composed of molecules that are fat-like in their composition. They're actually called lipids, so there's a little bit more to the molecule, but the tails of these lipids are fat molecules. So here we see an important concept, that molecules can self-assemble into larger, more complicated structures. Fat molecules can assemble into membrane-like structures. All cells have membranes. They perform an important function. They are the boundary of the living cell, keeping things inside the cell, keeping things outside the cell out, but allowing some things in and some things out. Now, in the rest of this drawing, let's see how these molecules that we've shown over here are related to the cell as pictured over here. Well, we've seen that fat molecules can assemble into membranes, and again the idea is fat molecules produced in these little chambers might have been forming membrane bound structures in these chambers, and if any of the chemistry was happening inside those membrane bound structures, then we have the beginnings of some kind of primitive cell type thing. Well, what about glucose? Yes, this would have been a sugar capable of being broken down for energy. Amino acids, we said, were the building blocks for protein, and nucleic acids the building blocks for DNA and RNA. Now, over here in this cell, let's take a look at the relationships between these three entities here. Let's take energy. Now, we know all living things have to derive energy. They have to have a source of energy. So, for example, amoeba eats paramecium to get energy. So organisms have to extract energy from their environment. Why does a cell need energy? Well, you have to make things in the cell. For example, making proteins. To make proteins requires energy. And how are proteins related to energy? Well, proteins are the ones that are performing the chemical reactions to extract the energy from food. For example, digestive proteins break down food, other proteins break down glucose, etc. So proteins are the workers in the cell, and they're going to be involved in extracting energy from food. Why do you need energy? To make the very proteins that you need to get energy. Well, where do these proteins come from? And here we have the other important part of the cell, the genetic replicator. You've got to have some kind of a molecule that specifies the design plan for making proteins. And DNA is that molecule in life today. So DNA stores the recipe to make the very proteins that are running cell activities. How are proteins related to DNA? We will see that proteins are needed to access that information and indeed to replicate the DNA so that the entire cell can reproduce. So if we think about a cell, then we can see three main functional parts of a cell. We have an outer boundary, and that's important because you have to keep all the chemistry going on in this compartment. If you rupture this membrane and the cytoplasm leaks out, the chemistry of life can't happen because the molecules will float away from each other. Right? So the boundary for life is critical. In addition, you have to have an engine, a metabolic engine, some capacity to extract energy from your food. And for living cells on Earth, proteins are the ones that do this. And you have to have a computer. Now, that analogy here is meant to mean that you've got to have some way of regulating the activities within the cell. So, for example, we'll see that DNA will control which proteins get made and when, And of course, then DNA is going to have the information to make those proteins, as well as be able to be duplicated so the entire cell can reproduce. So we've got membranes, we've got a metabolic engine, and we've got some computer, the hereditary information for this life. And finally, let me show you that picture in 1 second. Yeah, this was the drawing in the previous lesson where we have the three sort of functional components of a cell. A membrane boundary, the metabolic engine doing chemistry to get energy from food and energy to build stuff, and then the computer, DNA serving as the computer. And one last thing to note about this diagram. Over on the left, this whole part of the diagram, we could call this geochemistry. This is chemistry going on in the early Earth. On the right, within cell membranes, we have biochemistry. So what's happening here in the origin of life then, we have life emerging out of biochemistry, which itself, right, was emerging out of geochemistry. It's an incredible idea that chemistry might have produced the first living cells. And so in this final slide then, we can imagine at some point long ago, 4 billion years ago perhaps, or around that time, in these underwater alkaline hydrothermal vents, in one of the billions of chambers that might have been down there in the bottom of the ocean floor, chemistry had been going on for a long, long time. Membranes had been built. Simple proteins had been made. Perhaps a genetic molecule like RNA was synthesized, or DNA after that. And that this little structure was released into the ocean, into the environment. This would have been the birth of Luca, the last universal common ancestor, originating out of its rocky nursery here, to float off into the ocean, absorbing nutrients from the ocean, but setting the stage for the future evolution of all life on Earth.